Hello, everybody. For this lecture, we're going to discuss the anatomy of the nervous system, systems, cells, and cells that make up your nervous system. Okay, so first of all, we're going to start off with the nervous system. Okay, the central nervous system, or the CNS, is made up of the brain and the spinal cord. Okay, so everything outside of the brain and the spinal cord is then going to be the peripheral nervous system. Okay, so again, central nervous system, CNS, includes the brain and spinal cord. The brain is enclosed in the skull. The spinal cord is protected by the spine, the vertebrae. Okay, so everything outside of the CNS then is going to be considered the PNS, or the peripheral nervous system. Okay. And it serves to bring information into the CNS and carry signals out of the CNS. Okay. So if we look in a little bit more detail into the peripheral nervous system, you'll notice that it is broken down into two more parts. Okay, so the overall nervous system broken down into the CNS, central nervous system, and peripheral nervous system, and now the peripheral nervous system broken down into somatic nervous system and autonomic nervous system. So the word soma stands for body. So this would be the bodily nervous system. It consists of afferent and efferent nerves. Best way to know the difference here is that afferent, is the same as A in arrive. Okay, so they carry neural signals to the brain and spinal cord from the sensory stimuli. So these neural signals arrive at the brain and the spinal cord, so they're afferent. The efferent nerves exit. Okay, so they carry neural signals from the CNS to muscles and glands and so on. The autonomic nervous system is then broken down into two more parts, the sympathetic and the parasympathetic, and both of these are efferent. So here are those two systems. Again, autonomic nervous system broken down into sympathetic nervous system and then parasympathetic nervous system. For the sympathetic nervous system, just remember the fight or flight response. Okay, this is the system that gets us ready, aroused and able to take on those elements out in our environment that might cause fear or anything like that. Okay, they come out of the spinal cord and in the thoracic and the lumbar areas, whereas the parasympathetic nervous system, okay, is the rest and restore system, and it comes out of the cranial and sacral areas. Okay. So let's look at these in a little bit more detail. Okay, on the left-hand side, you have the sympathetic nervous system. On the right-hand side, you have the parasympathetic nervous system. Notice where they're stemming from. So the sympathetic nervous system is from the thoracic and lumbar areas. The parasympathetic is from the cranial and the sacral areas. And then you can see how they work on all these different internal organs. So the sympathetic nervous system speeding things up, getting things ready for action. The parasympathetic nervous system slowing things down and calming us down. So here are the major divisions of the nervous system all in one graphic for you. Okay, so the nervous system, again, broken down into central nervous system and peripheral nervous system. The central nervous system is just the brain or spinal cord. And so everything else aside from brain and spinal cord is peripheral nervous system. And then that's broken down into the somatic, the autonomic, and then the autonomic is again broken down into sympathetic and parasympathetic. Okay, now let's talk about the anatomy of neurons, which are specialized cells for the reception, conduction, and transmission of electrochemical signals. There are many different sizes and shapes of neurons. Okay, so in this graphic display here, it's showing you a unipolar, a bipolar, a multipolar, and then the interneuron on the far right. Most of the neurons that we study in this class, most of the neurons that are presented to you in graphic displays are going to be the multipolar neuron. Here's a way of thinking of a specific neuron as well, okay? So up at the very, very top, you have these dendrites, okay? These dendrites are in the input zone, okay? They collect and process information either from the environment or from other cells. And then you have the integration zone, and that's going to be the cell body, and that area that has the circle around there, that's the axon hillock there. That's the integration zone where the decision to produce a neurosignal is made. Then the neurosignal is transmitted away from the cell body by the axon, okay, which is the conduction zone where information can be electrically transmitted over great distances. And then finally, you have the output zone where the neuron transfers information to other cells. Okay, so if we look at this again, we have dendrites, soma or cell body, axon, and then the axon terminals. Okay, input, integration, conduction, and output. So dendrites receive information, axons transmit that information. So here are the external features of a neuron, okay, in more detail. Again, you'll notice that we have here two multipolar neurons, okay? So this would be neuron A, and then this would be neuron B, okay? So this is a cell body of neuron A. The cell membrane, we'll talk about in just a little bit here. The dendrites, again, input, receiving information. Axon hillock, the integration. The axon, the transmission of the neural signal. Okay. Covered in myelin, which we'll talk about that in a little bit too. And then the little areas, the little segments between myelin are called nodes of Ranvier, okay, or Ranvier. And then you end with the axon buttons or terminals. Sometimes there's more than one name for these. So if you look it up in a book or a different book, sometimes you'll see axon bulbs, sometimes axon terminals, sometimes axon buttons. And then these neurons, see, they don't actually touch. There's a space between this axon button and this, this neuron B, which in this case would be the soma in this case right here, almost to out to the dendrite. Be hard to tell if it was actually the dendrite or the soma on this one. And so that space between the two neurons is called the synapse. Okay, the major internal features, a neuron is just like any other cell of the body. It has a nucleus, it has the mitochondria, it goes through the Krebs cycle for energy, all of that kind of stuff, okay? But the important thing for us is that we understand that at these axon terminals here, and that's what this picture is blown up here for, you'll see that these are synaptic vesicles. They're fluid-filled sacs that contain the neurotransmitters. And when those neurotransmitters hit this presynaptic neuron or membrane here, then they're going to be released into the extracellular fluid. Okay. All right. Now the cell membrane is a lipid bilayer. Now this cell membrane right here could be any part of the neuron. It could be part of the dendrite, the axon, the soma, anywhere on that neuron. It has this bilipid layer, and it's actually just a barrier, okay, for the cell, so that certain things can't get through the cell membrane. It's very tight, okay. The only way things can get in and out are through these channel proteins and then the signal proteins, and we'll talk more about those in a later chapter. Okay, so let's go through this whole process again and look at the flow of information. Again, we have a presynaptic neuron, so this might be neuron A, and then we have a postsynaptic neuron, neuron B, okay? Now, why is that? That's because the neural message is being transmitted from A to B. So A is going to be presynaptic before the synapse, and this is going to be neuron B will be postsynaptic after the synapse. Okay? Then if we blow this picture up a little bit more, we'll notice that here's the axon coming down. Here's the axon buttons or terminals. You'll notice that this one is actually near the soma. You'll notice that this one is on a dendrite, dendrite. Okay. Then if we blow this up a little bit more, then we'll see the presynaptic neuron here is in blue, postsynaptic neuron in yellow. We'll see those vesicles again, fluid-filled sacs full of neurotransmitter. They'll migrate to the presynaptic membrane here and then release the neurotransmitter. If we blow up this little portion again here, then you'll see the neurotransmitter coming into this space. Remember, the neurons do not touch. And then you'll notice also the neurotransmitters attaching to what we call receptor site, okay, on another dendrite. So again, presynaptic neuron, postsynaptic neuron. Okay, let's go on to now glial cells. Glial cells actually outnumber neurons 10 to 1. Okay? They support neurons. They will actually engulf dead neurons, which keeps things clean in the environment, which is good. But they also act like scaffolding to protect those neurons. And recent evidence for glial communication and modulatory effects of glia on neuronal communication. So there's a lot of change going on in this field. We learn things every day. And now we're seeing that glial cells could possibly actually be communicating much like a neuron. Okay. There are 4 classes of glial cells. You have your oligodendrocytes. I know that's quite a word there, but it is pronounced oligodendrocytes, and they create the myelin sheaths for the central nervous system. Then you have Schwann cells, which create the myelin sheaths for the peripheral nervous system. You have astrocytes, astro meaning star-shaped, and they're very large. And then microglia, which micro meaning very small. Okay? And these are involved in response to injury or disease. Now, a little side note for the CNS and PNS and the different types of glial cells. One oligodendrocytes will provide myelin for several neurons in the central nervous system. In the Schwann cells, they will only produce one myelin sheath per neuron. And we'll see this a little bit in a graphic. The idea, though, here is that this is probably contributing quite a bit to when we see a spinal cord injury. Okay, the spinal cord, remember, was part of the central nervous system. So when it's severed, it can't repair itself. It's very, very hard for it to repair itself. In the peripheral nervous system, you can cut a nerve and those nerves find their way back through. And oftentimes you can get the feeling back in a finger or something that's been reconnected. Okay, that's probably due to the difference between the oligodendrocyte and the Schwann cells. And so here is that picture for you. One oligodendrocyte, right? And it's providing several pieces of myelin on various neurons. Okay? Here you have one Schwann cell, and it's providing one myelin sheath on that axon. Okay? So the Schwann cells can actually act like a little tunnel to help those axons feed back through that tunnel and help those nerves to find their proper connection once again. Okay, some other cell terminology for you. Just wanted to put it all in one place. The myelin providing glia, okay, these are the ones we talked about. Just remember that oligodendrocytes are in the CNS, Schwann cells are in the PNS, and then also clusters of cell bodies are considered nuclei, okay, or a cell body would be nucleus, but the cluster of cell bodies would be nuclei. In the peripheral nervous system, a cluster of cells is what we call ganglia. Okay, so you'll see these terms used and they won't tell you per se which is which, you just have to know. And then bundles of axons are called tracts in the CNS, but nerves in the PNS. Okay, so if you cut into a nerve into your finger, you'll realize that that is part of the peripheral nervous system. But if you were to sever a part of the brain itself, where you have a bundle of axons, we would call that a tract. Okay, let's talk about neuroanatomical techniques. All right, so we have Golgi stains, and they actually visualize an individual neuron. We have Nissl stains, which stain cell bodies. Okay, so the Golgi entire neuron, Nissl just the cell body, and then electron microscopy, which provides information about the details of neuronal structure. So I'll show you a picture of each one of these. So here is your Golgi method, and you'll notice that it stained the entire neuron, cell body, dendrites, axon, etc. Here for the Nissl stain, you'll see that they're just little blue dots, okay? Because it's just staining the cell body, not the dendrites, not the axon. And then here is your electron micrograph. Okay? So you're looking at a cell body, this gray area right here, and then all of these are little axon buttons or axon terminals. Okay? Here's another misconception, is that when we show graphs of, like I said, two multipolar neurons, working with one another there, it's never that simple. Okay, there's thousands and thousands of interconnections, okay, between the neurons. So it looks much more like this, very, very busy, some of these. Okay, so let's do another little recap here. If we take the adult brain, which this is a very similar size, okay, of a human brain, and we take one neuron out and magnify it, it would look like this multipolar neuron. So that's perfect, and we've studied that, right? Then if we magnify, remember these are dendrites, soma, axon terminals. If we magnify that, you'll see the little axon bulbs on here. Magnify one axon bulb, and you'll see how that axon then doesn't touch, but influences another neuron. If this was purple here, presynaptic, the yellow is going to be postsynaptic. If this is an axon, the purple, this yellow part is probably a dendrite, but it doesn't have to be. It could be a dendrite, could be a soma, could be another axon. But then if we magnify that, then you'll see, again, presynaptic purple, postsynaptic yellow, and now you're looking at the axon terminal here, and then let's say a dendrite, okay? Blow that up a little bit more, now you're looking at the cell membrane. Remember, this was a bipolar membrane, okay? And that protects it, so things can't go in and out of this membrane, except for through these ion channels, these protein channels. Okay, so the last thing here is we're going to talk about tracing techniques. So tracing is when you take a tracer molecule and you inject it into the brain, and then you identify where it goes, okay? Where it's being absorbed and where it's transported to. So if it's an anterior grade tracer, okay, that means that you can place it into the brain, it will be picked up by a neuron and then sent away or projected away from an area, okay? So if you follow where it's being projected away, then that's forward, okay? So that would be anterior grade tracing. If I put the molecule in this spot, but it ends up over here, then that's telling me where it goes, okay? However, if I put the molecule into the system, okay, and I find out where it came from, so it works backwards, then it would be a retrograde trace, okay? So where did it come from, okay? All right,